Good morning. Today I'll be discussing on very important link reaction, that is the pyruvate dehydrogenase complex reaction. Pyruvate formed in the cytosol by glycolysis is transported into the mitochondria by specific carrier present in the mitochondrial membrane. And inside the mitochondria, it is oxidatively decarboxylated to acetyl coenzyme A by a multi-enzyme complex. So conversion of pyruvate to acetyl coenzyme A is known as oxidative decarboxylation, and the enzyme is pyruvate dehydrogenase complex, which is found only in the mitochondria, and high activities of PDH are found in cardiac muscles and kidney. This multi-enzyme complex is having three components, which three component enzymatic activity. One is the pyruvate decarboxylase or dehydrogenase. Second is the dehydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. And the cofactors or coenzymes are thiamine pyrophosphate, lipoamide, coenzyme A, FAD, and NAD. That's how there is a role of important four vitamins. One is the thiamine, riboflavin, niacin, and pantothenic acid, which are acting as a cofactor. So you can see here how the conversion of pyruvate to acetyl coenzyme A takes place. Pyruvate in the presence of enzyme pyruvate dehydrogenase, which leads to the release of carbon dioxide. In the presence of TPP, it is converted up into the hydroxyethyl thiamine pyrophosphate, which along with the oxidized lipoic acid converts up into the acetyl lipoamide, and the reaction is mediated by dihydrolipoyl transacetylase. And in this particular reaction, there is a release of thiamine pyrophosphate. Now it is all said to be recycled for reuse in the previous reaction. Then acetyl lipoamide, in the next step, it is converted up into the reduced lipoic acid, and the acetyl coenzyme A is released here. So this step is very, very important, because in this, along with the coenzyme A, acetyl lipoamide is converted up into the acetyl coenzyme A, and the reduced form of lipoic acid is detached. So, and this reduced lipoic acid again converted up into the oxidized lipoic acid in the presence of FAD. In this reaction, FAD is converted up into the FADH2. The enzyme is dihydrolipoyl dehydrogenase. Then FADH2 again revert back to FAD in the presence of NAD plus. So NAD converts up into the NAD2H. So in overall, if you look into the bioenergetics, there is a total gain of 1 NADH, which is equivalent to 2.5 ATPs. So this is the summary. Pyruvate converting to acetyl coenzyme A. Coenzyme A, carbon dioxide releases. NAD converts to NADH, and there is a benefit of 2.5 ATPs. Then regulation of PDH complex, the end product inhibition, which is the allosteric inhibition by acetyl coenzyme A. Means if it is in sufficient quantity, so it will allosterically inhibit the PDH complex. Then NADH, abundance of NADH will also inhibit the reaction. Then covalent modification of PDH enzymes, that is via phosphorylation and dephosphorylation. So you can see it here in this particular diagram. So PDH complex B, which is an inactive phospho enzyme, and which is the it means in the phosphorylated form, this PDH complex is inactive. So in the presence of PDH phosphatase enzyme, it converts into the PDH complex, which is active dephosphorylated form. And this particular enzyme is being stimulated by calcium and magnesium ions. Insulin also favors the conversion of PDH complex into the active form, and further this PDH complex is converted up into the inactive form via the PDH kinases, which can be stimulated by NAD, ATP, NADH, acetyl coenzyme. So that means these are the inhibitors of PDH complex mediated reactions. And stimulators of this PDH complex are NAD plus, coenzyme A, pyruvate. So that's how we have seen how the inactivation and activation of PDH complex happen, and what are the activators and what are the inhibitors of the enzyme. So arsenic poisoning, the PDH is inhibited by the arsenic because arsenide binds to the thiol group of the lipoic acid and it becomes unavailable as a cofactor. Then PDH kinase, which is responsible for conversion of dephosphorylated form of PDH to phosphorylated form. So it is promoted by ATP, NADH, acetyl coenzyme A, and inhibited by NAD, coenzyme A, and pyruvate. It is very much apparent in this particular diagram. Then what are the clinical aspects? Thiamine deficiency, and that will lead to PDH activity, a decrease in the activity of PDH. Then pyruvate is converted to lactate. Instead of converting to acetyl coenzyme, it better converts to lactates, leading to lactic acidosis. Then inherited deficiency of glycolytic enzymes like pyruvate kinase, aldolase also hamper the formation of acetyl coenzyme right from pyruvate. Thank you.